Understanding biological functions requires investigation of their molecular compositions and dynamics at high spatial temporal resolution. Here's a schematic of a subcellular structure called the synaptic bouton, which makes up a neuronal connection in our brain. This rendered image contains 60 different synaptic proteins, occupying about 7% of its volume and constituting structures spanning nanometers to micrometers in size. When we zoom out, a single neuron can contain as many as 30,000 synaptic connections and extend millimeters, if not longer, in its full length. When we further zoom out, the human brain weighs about 1.4 kilograms and 15 centimeters in its length. It can contain up to 100 billion neurons interconnected with each other. The fundamental question my lab is pursuing is how can we identify and locate specific molecular species over such a broad range of length scales and biological complexity. So one approach my lab is exploring is a biochemical process that transforms biological specimens so that they can be studied more easily at higher resolutions. In this approach, cells and tissues are first fixed and chemically modified with a small molecule reagent. The modified specimens are then polymerized in situ to form a superabsorbent hydrogel composite. During this process, biomolecules of interest are anchored to the polymer chains. Finally, after digestion, the gel specimens are expanded in water, causing the anchored molecules to separate from each other. Because of the swelling effect, the biological samples are magnified physically so that a higher resolution can be achieved. So when we combine this biochemical process, with the latest microscopy techniques, we can start to capture the native molecular profiles of cells and tissues. For example, we have imaged at a nanoscopic resolution different neuronal structures using a fluorescence imaging technique called lattice light chain microscopy. This includes investigation of pre- and postsynaptic proteins in the mouse cortex, axons and their myelination profiles in the similar brain region, dopaminergic neurons and the presynaptic sites across the entire fly brain, as well as the correlation between axonal degeneration and certain disease markers in the human brain. Thanks for listening.